Hi, this presentation is made by Himanshu Dixit on the topic CRISPR good or bad. Clustered regularly interspaced short palindromic repeats, CRISPR, pronounced CRISPR. Prokaryotic DNA containing short repetitions of base sequences. Each repetition is followed by short segments of spacer DNA from previous exposures to a bacterial virus or plasmid. Advances in high-throughput technology like robotic automation has led to large-scale production of stem cell lines. This now brings stem cell technology into prime time for both academic and industry research centers. Combined with highly efficient gene editing techniques, here, CRISPR, we can now correct mutation in stem cell lines. Let us take the example of monogenic disorders like Alzheimer's disease, which we know there is high prevalence. Group with recent mutation in this disease, we can then create stem cells-derived neurons from the stem cells and use CRISPR to correct out or correct the mutation for personal and effectively creating a line from the same patient, with and without the disease mutation, are segments of genetic engineering v1.0. Limitation of this version are as follows. The DNA that could be manipulated was small. Random integration of foreign DNA. Multiple off-target effects. The techniques followed in genetic engineering v1.0 are as follows. Plasmid vector. Restriction endonuclease. DNA ligase. PCR. Sanger sequencing. Transgenic animals, plants. These techniques are not efficient and are impractical. Because of this, researchers started looking for different ways to engineer genomes, so they reached genetic engineering v2.0. Following techniques were used in genetic engineering v2.0. Homologous recombination. Artificial restriction enzyme. ZFN, zinc finger nuclease, complicated to design and suffer from having multiple off-target effects. TALEN, transcription activator like effector nuclease. CRISPR/Cas9. CRISPR, clustered regularly interspaced short palindromic repeats, began with exotic repeat sequence in bacterial genome, and it was originally considered exotic junk DNA with unknown function. Cas protein is the CRISPR-associated protein, is a family of genes associated with CRISPR that allows the interspace short palindromic repeats to be inserted or taken out of the gene. Function of the CRISPR is that it works as the bacterial immune system. In bacterial cell there is a phase double-stranded viral DNA which will be read by the Cas protein, and hence viral DNA will be inactivated using Cas protein, which will lead to the creation of a novel spacer. Buildup of these short spacer DNA from previous exposure to the virus allows the interspace palindromic repeats. It confers the immunity to the cell. Therefore, we basically have a machinery in which the cell can insert or remove the pieces of DNA sequence with very high precision and minimal off-target effects. Its advantage are as follows. Very affordable. Fast. High throughput friendly. High efficiency, up to 90%. Minimal off-target effects. One. Lab technicians inject a single sperm into an unfertilized egg. 2. The fertilized egg is injected with guide RNAs that target a specific gene and a template for the DNA cutting enzyme. 3. Researchers transfer healthy-looking embryos, now dividing into many cells into female monkey. Typically 3 embryos are transferred into surrogate. The twins Ming Ming and Ling Ling are born with multiple genetic changes, the first live primates created in experiment using CRISPR genome editing. Using CRISPR, germline modification will be possible. The germline modification will take 20-50 years to master, and it will be a topic of ethical debate. Genome editing might be used to model 3D facial shape from DNA. That's why companies like BGI is taking projects like Million Human Genomes. BGI is using this project to sequence 2,200 samples of genes of individual with high IQ in order to identify the genes associated with high IQ. The project is sequencing the genomes of people with IQ of 160 or higher. And the average Nobel laureate registers at around 145. These genome will be compared with sequences from general population in hope of identifying genes linked to higher IQ. The results for the same can be determined within 3 months period. Today the side effects of genome editing are very hard to determine. The argument made was to start a dialogue on the use of CRISPR technology. Increase in cure of these diseases will lead to decreased number of genetic diversity. We will be in a difficult position. You have a disease and you have a potential cure for that disease. Would you not correct that disease simply because it will decrease the genetic biodiversity? Following this argument, suffering of others is actually needed in order to maintain the genetic diversity. This question is a starting point of debate over ethnicity of CRISPR. CRISPR is still at a very initial stage and many more developments will be possible in near future. Thank you.